So welcome everyone, good afternoon. On behalf of the Statistical Society of Australia, I would like to welcome you to this webinar on getting to know our cellmates, a practical approach to microbiome analysis through a biostatistician's lens organized by the Statistical Society's Biostatistics and Bioinformatics section. My name is Sabine Braat, I'm the co-chair of that section and your host for this webinar. I would like to begin by acknowledging the traditional custodians of the land on which we sit today and pay my respect to their elders past and present. I extend that respect to Aboriginal and Torres Strait Islander peoples here today. So before I introduce the speaker, I have some practical announcements. The webinar will last 1 to 1.5 hours. We will allow some time for questions during the presentation and also at the end. We encourage you to let us know any questions you may have during the presentation as soon as possible via the chat, and we will address your question as soon as possible during one of the breaks. I may ask you to clarify your question, or you can ask the question yourself, in which case you can simply unmute yourself and speak up. So during question time, you can also put your hand up and signal that you have a question. So now without further delay, I would like to introduce you to the speaker of today, Dr. Elasma Milanzi. So Elasma obtained her Doctor of Science in Mathematics with specialization in biostatistics of Hasselt in Belgium. Her areas of interest include methods and applications for mixed models, survival analysis, dimension reduction, and microbiome analysis. She currently does independent statistical consulting and training, is an honorary research fellow in biostatistics with the University of Melbourne, and heads the analytics and research team at the startup company Halogen Health. She became interested in microbiome analysis during her collaboration with the Allergy and Lung Health Unit of the School of Population and Global Health at the University of Melbourne. And now I will hand over to Elasma. So Elasma, please feel free to start the presentation, and we'll talk again soon.
Thank you, Sabine, and thank you everyone for making time to be with me during this presentation, and thanks to the Statistical Society of Australia for providing this opportunity to talk about a subject that's of one of my interests. So without further ado, let's get to know our cellmates, as we put it. So as Sabine initially mentioned, I became interested in microbiome analysis during my stay at the University of Melbourne, where I was working in the lung healthy and allergy unit. So today what I'm basically doing is sharing with you my journey of analyzing that data. So my outline is as follows. I'll just briefly say something about the background of microbiome analysis, after which I'll introduce the data or the project which I was working on. The examples that I will show in this talk are coming from that project unless otherwise stated. And then I will talk a little bit about the processing of the sequence reads. This is kind of my main part in this talk. That's why I put there a biostatistician lens, because this bit is usually done by bioinformatics. So I wanted to share more about how to go about it from somebody who doesn't have that kind of a background. And then we'll explore some hypothesis formulations that you can do in microbiome analysis. And then I'll actually take you through some of the data analysis that we did for the project. The focus will not be mainly on the technical aspects, rather it's the intuition behind the methods that I used in analyzing the data. So in brief, microbiome in general refers to a collective genome of bacteria, viruses, and fungi, but mostly when people say microbiome, they refer to bacteria. So we'll keep to that definition of microbiome, referring to bacteria. It's been well known that bacteria, there are some good and bad bacteria. And actually, at some point, it was believed that the cells of bacteria were 30 times more than our own cells. So we were more bacteria than human beings. But that has changed over time. Now it's believed that it's almost 1-to-1, although the bacteria are still winning. So I think it just makes sense to spend time to know what they do, how they affect how we live, and how we can interact with them to make our well-being better. So initially, a lot of studies were focused on the gut bacteria because they were shown to infer immunity, especially to newly born infants. But now you see that the research on microbiome has really gone viral. That would depend on what age you are, whether say it's trending, it's viral, or now that because all of us know what exponential distribution is, you could say that it's increasing exponentially. The main reason is that now there are technologies available to help do the gene sequencing for microbiome in a more cheaper way than it used to be. The method that is now commonly used is called the amplicon gene sequencing. The amplicon refers to amplifying some targeted regions of the genes. And this is, if you contrast it to the well-known other method of shotgun, which performs like a broad survey of all the gene content. So the advantage of this is that we get a lot of data from one sample, what they call as the depth. The depth is higher than in the shotgun methods. But it comes with a catch because it's very error prone, because it has to go a few stages of sequencing, and it means each stage has a potential to introduce some errors in the gene sequencing process. So it makes it a bit more complicated, if I should use that word, to analyze the data. So maybe to give you a better perspective of what I'm talking about, I found this nice diagram that illustrates the processing pipeline. So in the first step here, we obtain a DNA sample. So that could be like a poo or a swab from the mouth. And then the bioinformaticians do the magic with that DNA, which I will not go into. And then they spit out these letters, which we are going to call them sequences, row-rich sequences. The idea is that we should be able to identify these sequences as genes for particular bacteria or species of bacteria. So my focus in this talk will be to see how we move from this bit where we get the raw sequences to the point where we are able to identify what kind of bacteria they are. So as I said before, if the amplicon sequencing was a flawless process, all we need to do is that once we get this, then we'll be happy to just identify them straight away. But the problem is that not all of the sequences that we get there are valid or are correct, or some of them just have poor quality. So that means before we can infer from the sequences we get from here, we have to make sure that they don't have any errors in them, or they're of good quality, because as you know, with data, it's garbage in, garbage out. So we want to make sure that we have the best possible data. Otherwise, we'll just be concluding that, okay, this region has this sort of bacteria, but in real sense, that is not the case. So it is this process of removing the sequencing errors to be able to make inference from valid data that I'm going to focus on. The reason I'm focusing on this is because as a biostatistician with a statistical background, I'm more used to data, well-structured data in a tabular form. So when I saw this kind of data and didn't actually know what was happening, it made me curious to actually understand it better. So it's not yet my playground. So if at any point I say anything that resembles to telling people to inject disinfectant in themselves, please raise your hand and let me know. But I'll try to portray this from a biostatistician point of view, because when we are analyzing data, we try to understand what the method is doing, like where the estimates are coming from. It makes it easier to spot the error or to interpret the results and put the things in context. So I think at this point, it will be good to introduce you to the project that I was working on. It comes from a cohort study hosted in Melbourne School of Population and Global Health. It recruited pregnant mothers during the antenatal care, specifically for families with history of allergic diseases, and they obtained breast milk samples from the mothers. It was at 3 days, which was basically colostrum, 5 days, and at 3 months. And the children of these mothers are the ones that have been followed for a long time. Actually, they are still being followed now. They should be around 25 years or so. The general question for collecting these milk samples was to try and find out whether the immunity acquired during breastfeeding reduced the risk of allergic diseases later in time. So there were a few approaches we could use to answer that question, and the microbiome composition was just one of the ways to do that. So the specific question I was looking at was to see whether there was evidence that the composition of microbiome in milk for mothers whose children developed atopic or clinical allergies was different. So you'll notice that if you compare with many microbiome studies, they collect the microbiome in 2 non-different groups. So it will be like diabetes versus no diabetes, and then they try to compare the microbiome between the 2 groups. But in our case, we collected the microbiome samples area, and the outcomes happen in the future. So it's slightly different from what many studies do, but that shouldn't be a big problem. And at this point, I should thank the RJ and Lung Health Unit for allowing me to use the data for this talk. And I should also be noted that all the results I'll show here have not yet been published. So it should not be shared at all. All right, so now let's go to the actual microbiome analysis. So I will strictly be following the tutorial that is presented on this website using the data tool package. It's a very easy to follow tutorial. The only problem that I had with it was I didn't actually know what every step was doing. Like I could get the results and everything, but I didn't know what to do with the results. So I will focus more on now my understanding of what every step does in that process and how we can interpret the results that we get from that process. So you can easily follow the code for doing this kind of analysis on that website. As such, I will not focus on the R code itself. So how does a raw sequence look like? Well, it usually have 4 lines. The first one is an identifier line, it identifies the sample. And then we have the sequence read itself, then a plus character and a quality score. An example of the sequence in our study is being shown on your screen now. So this line is the first line, that's the identifier. So we are looking at the sequence read from this ID, and you see that there is another number here, and that's because, as I said before, the apricot sequencing, the advantage is that it collects a lot of data from one sample. So this shows that this was the 539th sample from this individual, and usually they are in thousands. And the rest of it is a machine-specific identifier. So different technologies will have different characters on this side. But one thing that might be common to all is this number, the 1, which signifies that this was a paired sequence. So there's another sequence very similar to this, but with the 2 here to show that this one is a forward sequence and the other one is a reverse sequence. So when you're doing the analysis with the data tool in R, it requires that we should remove everything in this line, mainly so that it doesn't interfere with the actual sequence. So we should have something like this that's empty here, and it has only the ID remaining, and the sequence at the second line at the plus, and the last bit here is the quality of the sequence. So this is what the machine has assessed to be the quality of the sequence at that point. Before I continue, I should note that there is an N here, and we are not supposed to have that. So that's some of the data cleaning stuff we have to do with this kind of sequences, because we know that the valid bases are just C, G, A, and T. So before we can perform any other analysis, we have to make sure that our sequences only have the valid characters as well. I mostly used the Unix commands. I kind of abuse the grep command in Unix, but there are other options. I think Python is best suited for dealing with such kind of things. So as I was saying before, the last bit here, which is the 4th line, represent the quality of the sequence, the quality score of each measure. So for each character down here, there is a sequence base corresponding to it. So for example, if I look at E here, it refers to the quality of the T base there at the end. Each of these characters has a number associated with it. And for this, the machine that was used for this, they range from 0 to 40, 40 being a very good quality. So what it essentially relates to is the probability that this base was assigned to that position in error. So it's sometimes called the error rate. We'll see in a short while that this is very informative in like one of the steps to try and get rid of the sequencing errors from the raw sequence reads. So this is just what I was talking about, the quality score. So the types of errors that we can get from sequencing include just poor quality. So maybe the machine is not good enough, or the sample was contaminated, or it could just be some repetitive sequences, like they are in more abundance than they should be, or they could just be some sequences that should not be there at all. So this process, before we infer our sequences to find out what kind of bacteria they are, tries to remove as much of these errors as possible. So the first step when we get our sequences, so I'm assuming we've been able to import them to R and everything, is to visually look at their quality, because that's like the first step to see how many errors we have or what's the quality of our data. So I just took a sample of a few reads and plotted the quality. I find it good to look at the forward and reverse, the reverse sequences together. And again, this stage is like very important because it impacts the other stages. If we just ignore it and perhaps just take the default parameters, they might not be suitable for our data. So anyway, what's the graph showing? So the five pieces of information. Let's start with the heat map, so the grayish area. That tells you the frequency of the score at that position. So if we take at position 0, then this gray bit there represents the frequency of the 10 score for quality. And the orange lines there represents the quartiles. So the 25th, the median, and the 75th percentile. So for example, at base 200, the upper 75% of the score of the sequences had a score close to 40, which was good. And the green line there is the main score. So what you can see from our data here is that the quality declines very rapidly. Like when you see towards the end there, it goes close to 10, which is, I mean, in relative terms, it's a bad quality. So a few things we can take from here is that we should try and truncate our sequences at this point, say at 200, so that we don't have to deal with this part, which seems to have a lot of errors. And the other thing we can get from here is how strict we should be with the filtering, because after you have observed the quality of your data, the quality of your sequencing, the next step that comes through is to filter or to trim. So we can filter the sequences based on the maximum expected errors or on the quality of the samples, like what I was just saying, to truncate them at 200, or you can just set a number that any sequence that has a quality less than 2 or less than 10, we are going to discard it. So I think this stage is very important, in a sense that it paves the way to remove a lot of errors that might otherwise interfere with the data. The method, the data tool, says that it should not be a big problem if you leave the errors there. The reason being, the next step, which also tries to minimize the errors, uses the quality scores to determine which sequences are errors. But what I noted is that if you don't explore what parameters are good for your data, you end up, for example, having very few sequences going past this stage, or you end up with only a few sequences being identified at the end. So even if the method says it uses quality scores, I think it's important to try and figure out what works for the data. So just to give you an idea, I practically spent like the whole day trying to figure out which parameters are good for our data. Perhaps we can actually learn the lesson from the robot scandal, that we shouldn't leave everything to the machine. This is one of those situations where we have to have human input. So anyway, just try out what parameters suits the data, and then can proceed from there. So the next step in the data two workflow is to estimate the error rates from the data. So as I said before, the machine based quality scores are good. They are a good place to start, but it has been noted that they are not always in line with the observed errors in the data. So we have to try and find out what the actual errors in the data are. So to illustrate how these errors are calculated, I've given here some dummy sequences. So 2 sequences. So let's say this is the reference sequence. We want to calculate the error rate for the sequence below here. You'll notice that there are 2 places, the ones I have highlighted, that are different, that have different sequences. So if we first calculate what are called transitional probabilities. In loose terms, that means what's the probability that this A can be substituted by G. So what's the probability that A will change into G, and what's the probability that C will change into G? So you'll notice that if we get the transition probability for A to G and C to G and multiply them, what we are essentially trying to say is that what's the probability that this sequence is going to change into the reference sequence. So in other words, we're trying to see that what's the probability that this sequence is just an error, otherwise it should have been this sequence. Maybe to drive the point home, if we assume the extremes of probability, so say the error was 1 here for both of them, that means the A can for sure change into G, then it means we technically just have one sequence. But if the errors are 0, then we'll have 2 separate sequences. So that's the first step. The second step then tries to, before I go to the second step, I'll just show what the transitional probabilities looks like. So this is the graph that you get after you have estimated the error rates from the data. Sorry, I couldn't get these warnings off, but it just refers to these lines, the diagonal ones that it was going to infinite values. But what we are looking for in this graph is, first of all, the red line there represents the theoretical or the machine-based quality scores. So it's plotting the error frequency and the quality. So you can see that the machine-based are really not in line with the observed ones, which is what was noted, like the motivation for trying to estimate the error rates from the data. But what we should see here is that the frequency of errors decline with good quality. So the better the quality, the less the errors, and that the model-based errors that have been estimated should fit well with the observed error rates. So these are the transitional probabilities I was talking about. So the probability of A to C or A to G, it's usually, as you can see, a 4x4 matrix. But when calculating the actual error rates, the model incorporates the machine-based quality scores, which improve the estimation of these error rates. All right, so 2 points to get from this is, first of all, the model-based error rates should fit very well with the observed errors. And second, the frequency of errors should decrease with the quality score. If you find that something is not right, like for example, the error frequencies is increasing with the quality score, then it's better to check your data, whether you have removed all the headers, whether you have bases there that they shouldn't be there. And you can go and look again at the filter and trim stage to see if you were a bit more strict or if you should be strict. So those are the things that you should check when you see something is not going on. But usually with good data, that should be the trend. All right, so after estimating the error rates from the model, the second piece of information that's estimated is called the abundance p-value. So all these pieces of information, all these steps we're doing, the end goal is to try and eliminate the sequencing errors from our reads, our raw sequence. So at the moment, I'm just trying to go through the steps that this method goes through to try and achieve that. So the abundance p-value tries to estimate whether the abundance of a given sequence is consistent with the error rates observed in the data. So the idea is that the abundance of the sequences, so how many sequences, how many of a particular sequences we have in a sample, the assumption is that the abundance follows a Poisson distribution where the expected value is just the product of the error rate that we calculated previously and the total number of sequences in that sample. So what this p-value is trying to say is that, okay, if that's the expected value of the abundance, have we observed something extreme for that sequence? Have we observed something that shouldn't be observed under the null model? The null model being the assumption that the expected abundance is the error rate times the total number of samples. So in simple terms, it just means when the p-values are low, then it means the abundance of that sequence is higher than expected. So after we have had those two pieces of information, now it's time to come to eliminate the sequence errors, and find the unique sequences that are in that sample. So this is an overly simplistic way that this is done. That's my attempt to not go very technical. But what I'm trying to say here is that let's take this box number 1 to be, again, these are dummy sequences, not from our data, but let's take this to be all the sequences in a given sample. And then the one I have underlined here is the sequence with the most abundant sequence, and it's being used as a reference sequence. So the next step is to calculate the error rates, so those transition probabilities between each one of these sequences and the reference value. So we'll have the error rate for each of them. And based on those error rates, we can calculate their abundance p-value. And then we compare the p-values of all of them to a certain given threshold. So that's another thing you can look into the code, whether the default p-value given is something that you are comfortable with, but usually it's a very small value because it takes into account that you're doing a lot of multiple testing. So after calculating the abundance p-values, let's say the one highlighted in yellow had the smallest p-value. So what that means is it's more abundant than what the error rate in this partition or in this sample is suggesting. So in the next step, we take away that sequence and put it in a different partition where it now becomes the reference sequence. And then it means in the sequence 1, we only now remain with the reference sequence. The other remaining sequences, based on we calculate the lambdas again, based on both reference values. And then we assign them to the one that shows that it will have a higher abundance. So the ones that have a higher abundance when compared to this will go in this partition, and the other ones will go in these partitions. in very loose terms, which means don't exactly quote me on that, but what this algorithm is trying to do is to compare whether each one of these is very similar to the reference value. The one with the smallest p-value, it means it has a low error rate, and it's not very similar to the reference value. That's why it goes into this. And then we assess the remaining sequences and assign them to the sequences where we think they are very similar to. So it's a form of clustering, but based on the error rates and the abundance p-value. So at the end of the day, if, for example, we do the whole process again and we find that there's no p-value that is lower than the threshold, then these two are going to be our final unique sequences in our sample, and we'll assume that it has the abundance of 2, and here it has the abundance of 3. And then this is what is called the OTU table, or the operational taxonomic unit. That's because bacteria are not said to have species per se, but this is referred to as an OTU, and this is the abundance, and that's what's called the abundance table. Sorry, the OTU table. So an example of the results we got from all these processes are this. So the OTU ID here represents a particular sequence, and these are the samples, and the numbers represent the abundance of that sequence. And this is the taxonomic table, which represents the classification of each of those ETUs, like what family of bacteria they belong to. So in a nutshell, that's how the data to process flow work from in eliminating the errors from the sequencing. So I guess that would be the right place to stop and see if there are any other questions.
Yeah, thank you, Elasma. Yeah, we would like to take this little break to give people the opportunity to ask questions. So you're welcome to raise your hand and we can unmute you, or you can unmute yourself. And if you do ask a question, please state your name. At the moment, we haven't received any questions via the chat box. So I'll give people just a minute or so to give them the opportunity to ask a question if they have one at this stage. Okay. So I'll just give people a few more seconds in case there are any issues with unmuting. Okay. So what I'll do is, yes, yeah, go.
I have a question, if that's okay. It might be a really, really stupid question. Thanks so much for this, Elasma. I really don't know this area at all. So this has all been really, really helpful for me to understand a little bit more, because I know a lot of my colleagues and collaborators talk about this type of work, and it's just not something in my statistical work that I have to deal with. I was just really interested, like, obviously you're saying there's lots of errors that are introduced, which sounds understandable, but I guess it's at the point where you showed that the sequence codes that should only have these 4 letters can sometimes have an additional letter. And I was just, it seems like a really interesting error, because I guess I would have thought that the bioinformatics and the machine processing it would ensure that the sequence codes could only be the the 4 letters that they should be. And so this, excuse this incredibly ignorant question, but I was just wondering how that error is introduced, or where that error is introduced. So how can you get an N that appears there if it should always just be these CGAT?
Yeah, well, I wouldn't know the exact answer to that question, but I mean, because it's not entirely the machine that does the processing.
Okay.
Humans are involved as well, so it could be a real error when just transferring the sequences. So I'm not completely sure though.
No, yeah, that's the thing. I was wondering if you would be or not, but yeah, I think it's an interesting one, because I guess it was an error that I probably was naively thinking wouldn't be there. So it's really interesting for you to highlight the extent of the problems that you might be faced with if you deal with this kind of data. So thank you. Thanks for that.
Yeah, no, it's actually the other error was that it mistook the IDs of the samples. So like we had IDs in the sequences that we didn't have in the original samples that we had. So it's really good to kind of do a lot of intensive data cleaning to make sure I have clean data.
Absolutely. Yeah, I think you're just highlighting, just like, as you say, how important it is to really assess your data before you do anything with it, which I know as statisticians we should be aware of. But yeah, as I say, some of these issues I just didn't think were as big as they may be within this type of data. So thank you.
Yes, yeah. No worries.
Yeah, so I can answer the N question. So essentially, when the machine's reading the bases off, it's a fluorescent base with different coloured dyes for each one of the base, and it's when the machine can't tell whether it's an A, which is one colour, or a T. So it puts an N in and says that we can't. There's no differentiation between it. There's no.
Okay.
So, and that goes with the probability as well, with the calling of it as well. It's how confident is the peak. Essentially, you get a peak for a certain fluorophore colour.
Right.
And that's the confidence on that peak, on depending on how the other peaks for the other spectrums, so different wavelengths, so for each of the different colours.
Okay, great. Thank you. Thanks. That's really helpful.
Thank you.
Yeah, thank you, Karen and Nicolas. Before we move on, I have one question which I think might be helpful before we move on to the next part, Elasma. So someone asked if you could explain the nil model again. I think that was on the last slides about the abundance p-value.
Yeah.
Yeah.
This one?
Yes.
Yeah, okay. So the abundance p-value is just one of the pieces of information that are used later on to classify or to cluster similar sequences. So under the error model, we assume that the abundance of a given sequence has a Poisson distribution with the mean equal to the error rate, which we calculated before, and the number of sequences in that sample. So the p-value, the abundance p-value, tries to quantify the evidence that we are going to observe something abundance for that sequence that is extreme than what's assumed under the error rate model, where the expectation is the error rate times the number of sequences.
Okay, yeah, thank you. Yes, and if so if there are any further follow-up questions on that, we can talk about that after the presentation. But I hope that answered the question. So I propose we move on to the next part of the presentation. Thanks, Elasma.
All right, thank you. Right, so in the next part, I just want to talk a little bit about the possible hypotheses that you can have in analyzing microbiome data. So there's the concept of alpha diversity. This is the diversity within a sample. So how does the composition, how is the composition of microbiome within a given sample? Maybe just before I go on, the reason I wanted to talk a bit about this is because just like any other study, when we do a microbiome project, we need to specify like what's the primary analysis that we do. Because my observation so far has been that in a paper, for example, people do every possible analysis that we can do. I don't know if anyone has already quantified the impact that can have on the results, but I would assume the issues we have in other kind of studies of doing a lot of analysis would also transfer to the microbiome analysis. So in this section, I just wanted to highlight that there are different kind of hypotheses that we can look at. I don't think it's a crime to look at all of them, but when designing a study, I think we should have like a clear hypothesis of what we actually want to look at. So the first possibility is looking at alpha diversity, which looks at the composition within a sample. And this can be looked at based on the richness, which is just the number of species or the number of bacteria in one sample. And on the screen there, I'm showing 4 different samples. I borrowed this graph from somewhere on internet. I should reference it. But what's basically trying to say, if we are basing on alpha diversity on richness, what we are interested in is finding out which sample has the most species. So for example, here, sample 1 and sample 4 are the richest in terms of species. The other option is to look at relative abundance, which is called evenness. So like all the species around the same. So that's where the evenness comes from, that are they roughly around the same like in sample 4, or there's one dominating species in a particular sample. So we can either look at each one of them separately, or we can look at them together. Some of the common measures that are out there for alpha diversity is the CHA1, which is a richness based. So it really quantifies the expected number of species. And then there's the Simpson index and the Shannon index. Both of them, they quantify the alpha diversity, combining the richness and the evenness bit of it. But all the 3 measures are measuring the diversity within a sample. And then there's the beta diversity. So this compares the composition between 2 groups. So in the example here, sample 1 and sample 2 are in the same group, and sample 3 and sample 4 in a different group, treatment B. So we can take the mean of each species between the groups and obtain the different means for the treatment groups, and compare each. We can either compare each one of them individually, or we can compare the multivariate them together, like as a whole, treatment A versus treatment B. Which brings me to some of the popular methods that are out there for beta diversity. I think the very popular one is the weighted unifrag, which measures the proportion of shared branches on the tree. At this point, I should note that I didn't talk about a phylogenetic tree for our data, and that's because we haven't been able to obtain one because of memory issues with our data. It's too huge. So we weren't able to calculate such kind of measures that require a phylogenetic tree. But there's also another equivalent to MANOVA. This is a multivariate one, so it looks at the composition of one sample versus the composition of another sample as a whole, so not individual species. And similarly, there's a model based on the delete rate model. This is also a multivariate one, so it looks at the mean abundance vectors, whether they different between groups. I won't focus much on what each method does. It was just to throw in the methods that you can look more upon to do different, to answer questions that relate to different kind of hypotheses. So the other third kind of hypothesis is now isolating the different OTUs that are different in each of the samples. So this is a between the sample again kind of hypothesis. So the other common methods that we use for comparing means or for comparing abundances are used as well. So in this case, we're trying to find the actual species or the actual bacteria that are different between two groups. I think that's also like a popular form of analysis. And these are just some of the many methods that you can find there that try to look at the differential OTUs. That was a short section. Are there any questions at that point?
So again, please feel free to unmute yourself or to raise your hand. Maybe there are more questions now after this second part, and after giving it some more time while thinking about the first part. But if there are not any questions, then we can always move on to the last part, which is about analysis. So I'll give people another few seconds to see if there are any questions. Okay, maybe in view of time, Elasma, maybe let's move on to the last part.
Okay,
yeah.
Thank you. So for the data analysis, what I thought I would do is just try to show you the actual analysis of some of the methods that we did with our data. I created some sort of a Shiny app to do that. So I will just take you through some of those analyses, and we can see together the results that we got from our data. But before that, I just wanted to bring to your attention that the PhyloSeq package in R, or there's also a page for it, is a very useful starting point. That's where I think that's usually the starting point for analyzing microbiome data. So what it does is it allows you to put together the OTU table and the taxonomy table that we got after the denoising process, and the tree that we would eventually get, the phylogenetic tree that is, and the sample data. So like the characteristics of your samples. For example, in our case, our maternal characteristics, or the characteristics of the children, their RG outcomes. And the other thing is that there are two schools of thought there in regards to how to analyze microbiome data. I think the first one, and the one that most of us relate to, is to assume that the sequence reads that we get from a sample are just a random sample of all the possible sequences that can be obtained from that sample. So we can use the methods like the Pemanova, the Kruskal-Wallis, the methods that we are used to. But methods like the ANCOM here, they assume that the number of sequences that we get from a certain sample have an upper bound. So the instruments used for this sequencing can only deliver up to a certain number of sequences. So it's not a random sample as such, but it's compositional data because there's some limiting upper bound. I don't have strong opinion on that, but you can do a bit more of a research if you want to join one camp versus the other. But in both camps, there are methods that do all the possible hypotheses that you can formulate for microbiome data analysis. So at this point, I'll go to the I'll share with you the other screen, which is the R code or the app. So it's not a perfect app, but it can help you get started. It's on the GitHub. So if you just run the library Shiny and code the run GitHub, I'm not sure if you can see this clearly, but with my username and the directory, if I run that, it should bring me a window which has different kinds of analysis in the order which you should do them, really, because first of all, you need to get an overview of your data, which is data exploration, and then look at the different kind of hypotheses. I know I'm doing what I was just saying should not be done by looking at all possible analysis, but I think you bear with me. I was new to this topic, so I was trying all possible methods I could try. So how you can use this app is, first of all, you upload your data. So the PhyloSec project that I was talking about, where you combine the taxonomy table, the taxonomy table, the OTU table, and the sample data, and the tree if you have it. And then once you combine all that, then you can do almost all the models for analyzing microbiome data. And I'm planning to include a lot of other a lot of other methods in this, just if you want to get started on something and don't want to read it completely. So when it has finished uploading the data, it gives me the section here for the variables in the data set, and I have all the other information here. So at this point, for example, we're just looking at the relative abundance. So here I'm looking, for example, at the phylum level and seeing which bacteria are in high abundance overall in the population. So this is a combination of all samples from the women in this data. So as you can see here, that the proteobacteria was in high abundance, around like 30% median, followed by the actinobacteria. So I just did the graph so that they should give the top 4. And we can change, for example, if we want to look at the bar plot instead, and we can change which statistic, whether it should be based on the mean relative abundance or the median abundance. So this is just a bit exploring the data. Another way we can explore the data, and I should choose, let's say we should be interested in comparing our children who had eczema and at 1 year and those who didn't. We can do some pattern exploration as well. So this is the clustering methods. We're trying to see if there are any notable groups. So for example, here I have plotted the first 2 axes. And what I would be looking for in this graph is if there are any patterns in this regarding to eczema 1 and 2. I mean eczema 1 and 0, yes and no. So in this case, I really don't see any of those patterns. We can see the pattern in the data that there is one group here, one group there, and another one towards here, but not really something that relates to eczema. And we can do similar sorts of statistics, say summary statistics. So this would be the alpha diversity index, both the chow, Shannon, and inverse. So as we said before, chow is just the expected number of species. So here it says for those who did not have eczema, their expected number was about 45, expected number of species. Similar, we can do the similar interpretation for Shannon index and inverse Simpson. And we can also visualize this alpha diversity with graphs. And we can do a bit of the beta diversity as well. So comparing the whole composition of the composition of microbiome for mothers whose children had eczema at 1 year and those who did not, using the Pemanova method. It takes a bit of time. It should be there soon. There we go. So it gives just the F statistic. It should give a p-value somewhere, but it gets printed in the R console itself, so we can't see it here. But looking at the F statistics, we can see that there's no evidence of difference. So we could do all the statistics that I talked about, the methods I talked about. I'm happy to share with you the link to this app if you are interested. It's still way in development, but if you think you can find it useful, by all means, send me an email. I think I'll stop at that stage, Sabine. I don't know if there are any other questions.
Okay, yeah, thank you, Elasma. Yeah, we did have a question via the chat box, and I think it went back to the abundance p-value earlier on. So are you happy to answer that now, or would you like to first continue with finishing up this analysis part?
No, I think this is okay. That's all I wanted to show, so we can answer the abundance p-value.
Okay, that's good, yes. So it was a question about the p-value. So whether the p-value would mean that a particular sequence in abundance, sorry, I'm just going to read it out loud how it's stated. And Farah, please feel free to speak up if I'm misquoting your question. So it was a follow-up question on abundance p-value. So a lower p-value would mean that a particular sequence is in abundance and it can be clustered separately. Is that the understanding? Is that correct?
Yeah, that's correct. So it's more abundant than what the error model assumes. So it's relative to the null hypothesis, if I'm to put it like that. The null hypothesis being that the abundance of that species should be the error rate multiplied by the total number of samples. So if the p-value is low, it means that the abundance is higher than what that partition says or what that sample says.
Okay, yes, thank you. Farah, I hope it has answered your questions, and please feel free. Okay, yes, the question has been answered. Thanks, Elasma. So, and again, please feel free to mute yourself or raise your hand. We've reached the end of the presentation, so you're welcome to ask any questions you have at this stage. So I have maybe a more biostatistical question. So as a biostatistician, sample size calculations are an important part of my daily job. So I wonder, for microbiome data analysis, is there something like a sample size calculation?
Yeah, so that goes back to pre-formulating the hypothesis, so the research question, just as you do with any other study. So for example, if the hypothesis was to look at the alpha diversity, for example, to compare the alpha diversity between two groups. So say for alpha diversity in children with eczema and those without, then your outcome there is a continuous outcome. So you go about it the same way you go with a study that collects continuous data. On the other hand, if your primary analysis would be a better diversity outcome, say based on the multivariate method, then you would have to also calculate the sample size based on that outcome. For example, there is one that you can calculate if you want to analyze your data based on the maneuver method, which is a permutation maneuver. So it just goes back to knowing your pre-research question and then seeing which sample size calculation is possible. That's why it's good to have one pre-equation, and then you can base your sample sizes calculation on that.
Okay, yeah, thanks. Yeah. So just giving the opportunity for people to unmute themselves or to raise their hand. So I do have another question, Elasma. I'm sorry to go up. So again, in the biostatistics field, we have to deal with multiplicity, the increase of the false positive rate. So is that somehow in the microbiome analysis field?
Yeah, that's very much taken into account, especially when you're doing the differential OTUs analysis. So the p-value is usually corrected for multiple comparisons.
Yeah, okay, yes, yeah.
Can I ask another question, if that's okay?
Yeah.
Again, really stupid questions, Elasma. Thanks again for this. I don't know this field at all. So you were saying people decide in advance, obviously, or should decide in advance what question they want to ask, and that, I think, if I understand correctly, relates to whether they then will conduct the alpha diversity test or the beta diversity test, or this differential taxa, the differential OTUs. Is that correct? So the first thing is deciding which of those is most pertinent to what question you want to ask. Is that correct?
Yeah,
okay.
That's correct. Yeah.
And then within each of those, though, there are different options for what tests you can conduct to answer that question. Is there anything, any guidance really about which is the most appropriate test? I know you mentioned with one of them, obviously there's that assumption about whether it's from a random sample or not, but so is it really that assumption that determines which test you should use for all of these, or is there some other guidance as to what test to use, if that makes sense?
No, that was just one of them, like what you think, that was more about what you believe this data is.
Sure.
But as for the methods, each one of them has weaknesses and strengths. So for example, like the negative binomial model, I think assumes a negative correlation between the different OTUs, which is not always true. Okay. And then there are some methods which specifically take into account that the OTU is a space matrix, like it has a lot of zeros, and those methods might perform better than the ones that don't take that into account. Okay. So it's just a question of knowing which methods have stronger capabilities in regarding to your research question. So okay. There's no clear guidance, and of course the ease of doing the method as well, whether there's software available to do it.
Absolutely. So it's a bit of a pragmatic decision there too. Yeah.
The analysis.
Yeah. Okay. Great. Thanks, Elasma, and thanks for an excellent presentation. I certainly know a lot more than I did when I started this, so thank you.
Yeah, thank you for your time. And
we got a question via the chat box. So Alexandra has a question on, can you please explain again what the two schools of thoughts are related to the nature of the microbiome data?
Okay, so the first school of thought is the one that governs a lot of statistical questions, and that is if you take a sample, so a milk sample in this case, and do some gene sequencing on it, then it says the sequences that we get from that sample is just a random sample of all the possible sequences that we can get from that milk sample. So a random sample. The second school of thought is that the machine that was used to sequence that sample puts a limitation or an upper bound on how many sequences you can get from that sample. So it's not necessarily a random sample because it can't go beyond a certain number. So in that case, they think it's composition data. So it's relative to that upper bound. And is that clear?
Yeah, thanks, Elasma. Yes. Yes, Alexandra is happy with that. Thank you. Okay, so there's a lot of interest in the Shiny app. So I know people can contact you to get a link. So would you be happy to share your email address with the group for people that maybe to you by email to make any follow-up questions, as well as maybe getting a link to the Shiny app?
Yeah, that sounds good.
Okay. Yeah, we can follow up on that later after the meeting for people who are interested. And just looking at the chat box and giving people maybe a final opportunity to ask questions. Okay. No, I think that concludes the question round. So thank you again, Elasma. So I found it a very insightful presentation, and similar to Karen, as a biostatistician, this was all new to me and a very scary field to venture in if you're not familiar with microbiome. So thank you so much for this presentation.
Thank you for the opportunity.
Yeah. So I will thank everyone for joining this meeting. And also, I hope you all find it as helpful as I did. The Society has hosted a lot of events, and this was just one of the many webinars that we were hosting in June. But there will be more webinars like these in June and July, and I just want to draw your attention to 2 upcoming webinars/workshops. So there's one on the Friday, the 26th of June, on p-values and a normal assumption in scientific research by the president of the Statistical Society, Adrian Barnett. And I checked the website yesterday and there were a few places left, so be quick if you're interested. And the other one I want to draw your attention to was the mini conference, a virtual mini conference on the 9th of July, which is one jointly organized by the Statistical Society of Australia and the New Zealand Statistical Society, in place of the actual conference which was planned to happen that particular week. So if you're interested, please join one of those 2 workshops. Finally, if you want more information about the Statistical Society, you can have a look at the website. The web link is shown on the slide. And for those who joined this webinar and were not a member of the Statistical Society does webinars and workshops, but does a lot more than this. So feel free to browse around on our website and contact the Society, especially maybe consider if you would like to become a member. So I would like to thank you all again for attending the webinar, and this will now be the end of this webinar. Thank you again.